Hey guys, so today we're going to be doing a quick video on nitric oxide pathway inside of smooth muscle cells and how that leads to vasodilation. So a lot of you probably already know that nitric oxide is a potent vasodilator and it's used in vasodilation throughout the body. But we're going to be taking a specific case here and running through the pathway of how nitric oxide can become active and how this results in vasodilation. So let's say we're in an endothelial cell, okay? And we have nitric oxide being synthesized. Nitric oxide is synthesized by an enzyme, which we have labeled over here, called nitric oxide synthase, and I'm just going to zoom in for a second. So nitric oxide synthase is the enzyme that makes nitric oxide, and the main purpose of nitric oxide is to activate this enzyme over here in smooth muscle called myosin light chain phosphatase. So not kinase with a K, phosphatase, okay? If you remember from our smooth muscle video, myosin light chain kinase phosphorylates myosin, which allows for cross-bridge cycling in smooth muscle. Well, here myosin light chain phosphatase does the opposite and dephosphorylates the myosin light chain, which results in no cross-bridge cycling and vasodilation because actin and myosin cannot bind when myosin is dephosphorylated in smooth muscle. Okay, so let's take a hypothetical situation. Let's say we have an M1 or an M3 receptor. These are muscarinic receptors, and they act through the GQ pathway for G protein coupled receptors. And the GQ pathway is known to be able to activate nitric oxide synthase. Now, how does that happen? So acetylcholine is going to go ahead and bind our M1 or our M3 receptor. This receptor then activates its G protein coupled unit called GQ. GQ, as you know, has alpha, beta, gamma subunits. The alpha subunit detaches and is going to activate another enzyme called phospholipase C on the cell membrane. This phospholipase C then does our key reaction here and converts phosphatidylinositol bisphosphate, or PIP2, into inositol trisphosphate, IP3, plus diacylglycerol. What we really want here is our inositol trisphosphate, because that can then go inside the cell to the smooth ER, or endoplasmic reticulum, and open calcium channels, which then causes the release of calcium because it's going to flow out of the SER, high concentration to low concentration in the cytoplasm. Calcium will then bind to a protein called calmodulin and form a complex called the calcium-calmodulin complex. These two together now have the ability to activate nitric oxide synthase. Now, nitric oxide synthase has a particular reaction that it does in order to create nitric oxide, and I'll just go through some of the general details. Okay, so in order to create nitric oxide, you need three things. You need arginine, oxygen, and NADPH. Arginine, as you know, has a lot of nitrogens on it, on its side chain, so it's perfect for creating nitric oxide. Then we have our oxygen for our O group on our NO, and we have NADPH to provide a reducing agent. So arginine in this process gets converted to citrulline as it loses its nitrogen group. Oxygen gets put into the nitric oxide, and NADPH loses the H and gets converted to NADP+, so it gets reduced. Okay, so now we have our nitric oxide from our arginine to citrulline reaction. What's nitric oxide's purpose in the smooth muscle? Well, it goes to activate guanylyl cyclase. This guanylyl cyclase is going to take a GTP and convert it to cGMP. Then this cGMP, or cyclic guanylyl monophosphate, here's guanylyl triphosphate, or guanosine triphosphate. This is the same thing as, or very similar to adenosine triphosphate. So if you remember the adenylyl cyclase reaction, when we have a GS receptor, we have adenylyl cyclase converting ATP to cAMP. That's similar here. And then we have cGMP going to act on protein kinase G, which will then activate myosin light chain phosphatase. So it's going to phosphorylate because it's a kinase, and it's going to activate the phosphatase. This phosphatase is going to remove the phosphate off of our myosin light chain, or the myosin head, and now we're going to get vasodilation because our smooth muscle can no longer perform cross-bridge cycling or contract. So let's do a quick overview to make sure everything's in place. So our acetylcholine binds our muscarinic receptor. Our muscarinic receptor then activates GQ, G protein. The alpha unit from GQ activates phospholipase C. Phospholipase C converts PIP2 to IP3 in DAG. IP3 then goes to the sarcoplasmic reticulum, causes the release of calcium. Calcium binds calmodulin, forming a calcium calmodulin unit that activates nitric oxide synthase. Nitric oxide synthase then goes and activates guanylyl cyclase, which converts GTP to CGMP. CGMP can then act on protein kinase G, activating it, and protein kinase G activates myosin light chain phosphatase, which then causes vasodilation. Okay, so this isn't the only way nitric oxide synthase can be activated. There are different kinds of nitric oxide synthase, inducible, endothelial types, and they can be activated by things that usually cause inflammation. So if another activator would be an example. Another example would be thromboxane, if you wanted to activate it using inflammation or other inflammatory mediators. So those activate something called INOS, which is inducible nitric oxide synthase. Okay, so once you're at the NOS part of the pathway, the rest is generally the same. How you get there can vary. Here we have a muscarinic way to get there. In future videos, we'll do other ways of activating nitric oxide synthase. Alright, I hope this video helped, and I'll see you in the next one.